A warm welcome to our webinar series Phenomics again, now having the 6th version already of this webinar series, which we have established quite a while ago in compensation for the COVID-19 limitations. And we have, with that series, a kind of format which enables the collection of questions in advance, and then we have these Q&A sessions as we have it nowadays. The whole session is being organized by 3 partners: the International Plant Phenotyping Network, the European Infrastructure Emphasis, and the European project EPP in 2020. So my name being Sven Fahner, I'm based at the Institute of Plant Sciences at Forschungszentrum Jülich, and I will just moderate you through the session today. Obviously, this session is not about me, but about our special guest, Hela Chikh-Rouhou from Tunisia. We are very happy to have her join today. We have this video made available on the homepage, on this quite impressive video, which has been seen by a wide community, as we have already realized. So we are very much looking forward to go into that discussion, Hela, and it's great to have you here. Thanks for sharing your time with us. Yeah. A couple of words just what we're going to do today. So I will shortly introduce Hela in a second, and then she will have about 5, 6, 7 minutes time just to sum up her presentation that is on the video, just to give all the participants here a little bit of a reminder, and also to give some time if you want to think about also some new questions in addition to the ones we have already received. The way we do it is we would very much ask you to provide your questions in the chat if you have any, and then I will moderate this through. And this is the way we move forward for today. And this video obviously will be put online afterwards as well, so you have the possibility to watch it as well, and then come back to Hela to ask some further questions on this really interesting topic of hers. As I said, a very short introduction to Hela. So she's a researcher at the Regional Research Center on Horticulture and Organic Agriculture in Tunisia. She holds a PhD in genetics and plant breeding from the University of Zaragoza in Spain, and an agricultural engineer diploma in biotechnology and seed production from the National Agronomic Institute of Tunisia. Her main topic is work on the valorization and preservation of cucurbits' local genetic resources, like melons, as she will address today in that session, and their exploitation for sustainable agriculture. And she has been involved already in several national and international projects dealing with the exploration of genetic diversity in vegetable species using phenotypic and molecular marker approaches. The talk she's given is on regional melon crops in the Mediterranean basin, and they affected by pathogens and pests, and with the challenge of identifying and using resistant cultivars in that context. And I think it's not me moving forward here, but I would just now open the platform for Hela.
Hi.
So,
Thank
your
you.
input there.
Hello, Sven. Thank you. Thank you for giving me this opportunity. My talk will gonna be about the evaluation of melon landraces to biotic stress in order to start a breeding program, and in order to be used also as rootstock or maybe as resistance cultivars in the affected areas. I will show you later on the summary of the presentation, but just to say that I'm working especially on the valorization and the preservation of cucurbits, local landraces, for their sustainable use in the future. As we all know, we are facing climate change challenges, and we have to take aware of these problems, and we have to valorize more our landraces, because these landraces are very rich in gene of resistance, and we have to preserve them and to take care of our landraces, which is our identity also. So I'm currently managing a melon breeding program, as you mentioned, Sevan. And also I'm working on other cucurbits crops, such as watermelon Also, I'm working on pumpkins, sponge gourd, and battle gourd. I've started recently working on battle gourd and sponge gourd, and also evaluating the gene of the resistance to the biotic stress, especially. And that's it for this.
Great, thanks for that. And if you like to, I guess you could already share your screen and introduce us or summarize. Better things.
Summarize.
Yes, you had. Yeah, it's coming.
It's coming. Okay.
Okay, there we see your screen.
I will put. Okay.
Yes, there it is. Thanks, Hela.
So the title, as you all know. So just to start from the beginning, as I've said before, so the landraces are adapted to their growing conditions and represent an opportunity to explore their variability and to identify promising traits that could be exploited in melon breeding programs. And also in Tunisia, there is a lack of information regarding these melon landraces, and there is a little attention regarding its preservation. So in this context and from this problematic, I started my program on melon landraces. So as we all know, biotic stress is affecting all the crops, and for melon, fusarium wilt, it's a soil-borne disease, powdery mildew is an airborne disease, and the aphid, which is vector of many viruses, are affecting a lot the melon crops. And the genetic resistance is preferred control strategy because it is more safe for the environment and for the human health. And before starting, we have to know there is several resistance genes to fusarium, melonis, and to the aphid that have been genetically characterized. We have FOM1 gene, FOM2 gene, and the VAT gene. And each gene, it's a major gene conferring resistance to the race of the pathogen. Regarding powdery mildew, there is a controversy regarding the number of genes implicated in controlling the resistance to powdery mildew, and it is cited that there is different QTLs that is implicated in the control resistance of powdery mildew. So to start a melon breeding program to biotic stress, it includes 3 principal steps, which are screening germplasm collection, to study the mode of inheritance, and also to introgress this resistance trait into the elite cultivars. In our presentation, we're going to focus only on the first step, which is screening germplasm collection, in order to identify sources of resistance. And this can be done using phenotyping and molecular approach. This step is very crucial because if we don't phenotype it probably, we'll never find that candidate gene and more even the molecular markers linked to the candidate gene. So this first step is very crucial, and we have to do it as good as possible. For that reason, we do phenotyping and molecular approach in order to be sure of the genotype or the phenotype that we select. So our objective is to identify melon landraces resistant to the main fungal diseases that I have stated before. And if we found melon accession with multiple resistance, it will be good. But above all this is to obtain a detailed phenotypic characterization of these landraces in order to be used in breeding program and also for their conservation. Is like passport data for the landraces. In order, if we have to start another program or another evaluation, we have the data about these landraces, and just we take the landrace that we need for our programs. But before doing our breeding program for biotic stress, we have to know which race of the pathogen is in the area. For example, in Fusarium melonis, we have 4 races of the pathogen. Race 0, race 1, race 2, and race 1-2. The race 1-2 is the most virulent. So if we have to identify the races in the area, I cannot start a breeding program. I start a breeding program for race 0 and I don't have race in Tunisia. So we have to identify the races in order to start an appropriate breeding program. And here in Tunisia, we identify that 4 races are present in the different regions prospected here, with predominance of the race 1-2. This is the most virulent race. For the powdery mildew, also there is different races of the pathogen. And in the Mediterranean regions, races 1, 2, 5, and 3.5 are the most predominant. And here in Tunisia, in our experiments, we found that race 2 is the most predominant, but we need to confirm the results doing another experiment. As you know, we have to do the experiments twice and in different areas in order to be sure of the race exactly. So to do the screening, there is a lot of methodology used, but the inoculation, artificial inoculation, is the most used for the screening for resistance to Fusarium wilt. And this artificial inoculation is done by trimming the roots of the plants at the first true leaf stage, and then dipping these roots in the conidial suspension. And then we put the pots of the plants in controlled growth chamber, and in order to observe the symptoms of the pathogen on the plants. As we all know, the symptoms or the severity of the disease is influenced by the genotype of the plant, if it's resistant or susceptible, also of the environment where we put our plants to be evaluated, and also it depends of the virulence of the pathogen. So once we put the plants in growth chamber, we observe the symptoms and we evaluate if there is resistance or no. And regarding the 27 melon accession evaluated for races 0, 1, and 2, as shown here, we observed that several accessions were resistant to these races 0, 1, and 2, and mostly are qualitative. And as I've said before, for the qualitative resistance, it is controlled by a major gene. And for the case of 0, 1, and 2, we have 2 major genes, FOM1 gene and FOM2 gene, which confer resistance both to 0, 1 and 0, 2 respectively. For the resistance to the race 1.2, which is the most virulent race, and this resistance is also controlled by several genes and are recessive, but no molecular markers for that race is available until now. For the resistance to race 1.2, so we evaluated as it is quantitative resistance, so we evaluate the resistance using the area under the disease progress curve. It is quantitative parameter that allowed us to differentiate between the different accession of the tested accession of melon. And here, as shown in the histogram, we found that 5 or 6 accession showed high resistance to this race 1.2. For the screening to powdery mildew, we evaluate this under greenhouse conditions and a natural infection. And here we evaluated the symptoms of the powdery mildew, which is the powdery white powder, which is the mycelium of the pathogen that we can observe on the leaves. So here we observe the leaf coverage, and we evaluate the disease and this incidence for each accession. And here, as we can observe, for the disease incidence for the different melon accessions, there is a great distribution of the different accession. There is highly susceptible to highly resistant. And the most important thing is that 4 accessions were presented here in pink colors were highly resistant to the powdery mildew, and they show disease incidence not exceeding 10%. And those presented in blue colors present the accession that are moderately resistant to this pathogen. So this resistance, if identified for the powdery mildew, we need to characterize this resistance, because as I've said from the beginning, for the powdery mildew, there is a controversy regarding the number of genes implicated for this for this pathogen, and the resistance identified is race-specific in the accession where we identified the resistance for the pathogen in question. So we need to characterize this resistance in the Tunisian melon accession that we found by doing crosses between resistant and susceptible genotypes. For the aphid, which is vector of many pathogenic viruses, the main symptoms of the aphid is the curling. It causes curling in the leaves, as observed here, and also it colonizes the leaves, as we can observe here. Sometimes the colonization is very heavy, and sometimes we can observe a little aphids in the colonization. So the evaluation also here is done upon natural infestation under greenhouse condition. And the evaluation, as shown here, we evaluated the colonization and the curling of the leaves. And as we observed, large variability of response to the aphid, and 9 accessions that are presented here in pink color, they were tolerant to the aphid. They did not show curling symptoms. And these 9 accession tolerant may be resistant because they may have the VAD gene, this VAD gene, which confer resistance both to the aphid and also to the virus transmitted by this aphid. So it is very interesting if we find these VAD genes in these 9 accession identified as tolerant. And also the most interesting thing is 4 accessions are multi-resistant. They showed resistance to the aphid here, and also they showed the resistance to the powdery mildew in the other experiment. So molecular markers, this is, so I was talking now about the phenotyping done classical phenotyping. We observe the symptoms, we evaluate the reaction of the different plants to the pathogen, and we evaluate the reaction, and we classify if it's resistant, susceptible, or tolerant. And in order to be more precise and to make an accurate characterization of the germplasm, we need to use the tightly linked markers in order to be sure of our selection. Hopefully, tightly linked markers are available to the 3 major genes that I've told before, FOM1, FOM2, and VAD genes. The 3 major genes, there are tightly linked markers available, and we use them in order to evaluate our melon accessions. So regarding the linked markers used for F11 gene, we observed that 7 accessions were homozygous for the gene and 6 heterozygous for the gene. Regarding the FOM2 gene, we observed there is 2 accessions. They presented the gene in question. And for the VAD gene, this experiment was done in Spanish laboratory, and we identified in the 14 accession evaluated that the VAD gene is present only in 1 accession. So the most important thing here is when we identified molecularly the presence of the resistance gene in this accession or in the other, so we can pyramid these all resistance of gene in 1 accession. And this is the most important thing in a breeding program, is to pyramid all these genes in 1 accession. And also, as a breeder, I have to know more about the accession. It's good to know it's resistant or not, but also a well-characterized crop genetic resource is a precondition for effective breeding and also for genetic conservation. So in order to do the phenotypic characterization, several traits were evaluated regarding the flower, regarding the leaf, the fruit, if it's decent or not, if it has grooves or not, depth of the grooves, color, all these parameters are mentioned here in the table. A lot of traits are evaluated phenotypically and also for the seeds in order to make an appropriate characterization of these melon landraces that show the resistance or not to the pathogen. So here we observed large genetic diversity as shown here in the picture, great diversity for these melon landraces. And we did also the phenotypic dendrogram basic on quantitative and qualitative phenotypic traits in order to separate the different landraces in different groups. And also we did the phenotypic correlation, which will help the breeders in order to select the appropriate trait. So the knowledge of phenotypic variation and the traits relationship will assist the breeders to develop the most adaptive and productive cultivars. It's a very crucial step also. And also here for the phenotypic evaluation of the melon landraces, and for a more precise clustering, we also did a molecular analysis markers that we used in order to obtain a more precise clustering for these landraces. So as to summarize a little bit, so phenotyping tools in common use or the classical phenotyping is labor intensive because we have to go to the field, collect data, we have to make the experiments for several years in different regions. It's time consuming and also costly. And with coming of the high throughput phenotyping, we are going to help raise the precision, will reduce laboratory mechanization, and will help a lot the breeders in order to make the phenotyping. But unfortunately, these platforms are designed for a limited range of species and also for a limited range of countries. We don't all have these facilities to do the high throughput phenotyping. And the concluding remark is, as showed here, the importance of the local landraces as a valuable genetic reservoirs and the urgent need to plan strategy for its conservation and utilization in breeding programs. We also showed that several resistance accession identified were resistant to one pathogen or to several pathogens. And these resistant accession can be used directly by local farmers or also as sources of resistance in breeding program. And this is the objective of our research. And as we said before, plant breeding needs an accurate phenotyping is the most important thing. Phenotyping done by classical phenotyping or high throughput phenotyping, the most important is to do appropriately the phenotyping in order to select the appropriate and the resistant genotypes. And this can be greatly improved by integrating molecular markers. For us, as we use classical phenotyping in order to be more precise in our selection, so we integrated molecular markers for the genes that are, for the gene of resistance and for the molecular markers available for these resistance. Sometimes we don't have the molecular markers linked to the resistance gene and are not available. So we have to look for the molecular markers linked to the gene of resistance that we will identify in our accessions. And also it is important to do the phenotyping to abiotic stress, drought, and salinity tolerance is needed in order to develop varieties that are able to cope with the climate change challenges. And this is the most important things, I think. And this is the concluding remarks of my presentation. And that's it.
Right. Thank you very much for that summary, Hela. Very, very interesting, very helpful. I would just like also to encourage the audience here, if there are any further questions, just put them in the chat. And I will otherwise just bring forward the questions we had received in advance to the session today.
Okay.
Get the input and experience from Hela on these questions. Hela, the first one is a rather, a little bit of general one, addressing the melon accessions you had used for and you had investigated during your study. So could you share some insight why you chose these accessions exactly that you did use for the study, and maybe why you didn't choose other ones?
Okay. Great question. Yeah, I didn't choose from the beginning when I was starting my breeding program. I go to the genome bank to take some accession, but I didn't find. So I start from the beginning, doing by myself with our institution, doing collection and prospection from local farmers, and I started collecting different accession from local farmers. And unfortunately, very few farmers are still maintaining their local landraces. I was really surprised because I found that the farmers are using hybrids and commercial varieties that are higher yield and are more resistant. And we are facing now the genetic erosion. I selected these from all the countries, 27 or 15 accessions that I selected, and I started doing my program. So this accession collected from farmers. So I take it from them and I evaluate them. I evaluate this accession. I didn't select this accession, and I don't have any information regarding these accession before. So when I started my breeding program from the beginning, it was like that, collecting from farmers, taking seeds from farmers, and I started evaluating, doing experimental designs in order to evaluate their resistance and to evaluate their tolerance, and also to evaluate their agronomic and phenotypic traits. And it was not based on selection before. And I was interested in the land races, as I've said before, because the land races are our identity and we have to preserve them. From the beginning, it was selecting and to preserve our collection, to preserve our melon land races. And then I started doing research of evaluation to biotic stress, and maybe next time to abiotic stress, and doing research on that sense. Hope it's clear.
It is. Thanks, Hela. You raised this topic of identity trust and preserving the identity. Did you then ever consider having also land races beyond Tunisia, or is this something that you would consider for the future, or is this just not of specific interest, as you said, considering the local approach that you took?
From the moment, just local land races, to start to preserve them, to valorize them, and to use them for breeding program to enhance some traits, because not all the traits that, because you found sometimes that the accession is resistant to the pathogen, but the agronomically or fruit quality is not so good. So this is challenging for a breeder, because you have to make resistant accession, but also with the great yield, because it's what the farmer is looking for. It's yielding, and also with acceptable fruit quality. And sometimes we don't really found that. So we have, for now, just working on the local land races, and as I found some resistance genes in these accession, so I will work on that and will enhance the quality and the resistance of these local land races, and to give to the farmers later on with a good quality and with good yield, and also.
Right. Okay. We had one more specific question, which was about or addressing the greenhouse experiments you made regarding powdery mildew. And there have been a question regarding if there was some more information about the setup you had in terms of the growing time of the melons, as well as the repetitions and the number of accessions you had used. Could you provide some more information on that one?
Yeah. For the experimental design done under greenhouse condition, for all the breeders and for all the research, when we do an experimental design, we have to do the repetition. So we have to choose an experimental design, and the randomized complete block design is what I use usually with 3 replications. So I have 3 blocks with a different accession evaluated under greenhouse conditions. And I started evaluating the symptoms of the powdery mildew once the plants, because I put also susceptible controls. There is susceptible controls that are susceptible to the powdery mildew. And when this susceptible control is with a lot of symptoms with powdery mildew, I've started evaluating these accession. And I've said before, I have 3 blocks it's a randomized complete plot design with the different blocks. In these different blocks, there are many replications and different plants. And for each accession, we have minimum of 6 plants per each accession. So, and this experiment was done in 2 successive years because we have to confirm the results of the greenhouse condition also. And if we have a lot of accession in the greenhouse, so we do the experiment in 2 different greenhouses in order to evaluate more accession at the same time.
Yeah, right. Got that point. You already, there's another interesting topic, which is now, because you mentioned the role of farmers several times already by now. So how is, in practical terms, how are farmers involved in what you're doing? You mentioned already that obviously they help, or it's a support for you in terms of choosing the accessions. But how is it based on just personal contacts you would have and the experience you would have in dealing with them? Or is there some, I don't know, strategic structured approach? So how do you involve the farmers in your work that you do?
From the beginnings, when we started collecting seeds, so we have contacts. We cannot go directly to the farmers, but we have governmental instruction. And there is, for the Ministry of Agriculture here in Tunisia, there are regional, it's called CRDA. It's implicated in each region. It is it's in contact direct with the farmers. So as a researcher, I contact the CRDAs in order to make my contacts with the farmers. We cannot go there and they will not collaborate. And if it's a governmental it's initiation, governmental initiation, so they accept you and they collaborate. And I found really when I was collecting the local landraces, I was really shocked, as I've said before, because very few farmers are still maintaining their local landraces, not only for melon, for all the crops in Tunisia. Very few farmers are still maintaining their landraces. As I've said, they prefer to use the commercial varieties that are very highly yield, and this is what the farmer is looking for. Is money is for a variety that produce highly producer. So I started from that, and they were very collaborative because they were also expecting something. They was, yeah, we're gonna work with landraces. Yeah, we need our landraces, and now the farmers are more aware of this problematic. And if we lose these valuable landraces, we're gonna lose a lot of things. So they are more aware of that, but. No, we are facing genetic erosion of our landraces, unfortunately, and for that we have to actuate, and urgent things need to be done now before other things in order to preserve these landraces. So we collect them, so I collect these landraces, I evaluate them, but also I give those samples from these accession to the genome bank. There they will be in safe. So I gave them samples of all the accession that I have collected. So a collaboration with the genome bank is also done. And this is very important, I think, just to start a breeding program, but also to conserve. This is the most important, to conserve them and to valorize them later in what we need. So I take them from the farmers, and I give also to the genome bank to preserve them. They are well conserved there in good conditions.
Right. Yes, that's what I hope and expect. Talking a little bit about the pathogens and pests that you investigated. Looking a little bit into the future, and I think before we started this formal session here, we had already a little bit talked about the topic of climate change. Do you think there may be any changes in the pathogens and pests coming up that are of relevant for the different melons that you investigated. Is there a drift potentially that you may need to investigate other pathogens or other races of these pathogens you invested?
The most problematic things for melon crops is powdery mildew, is devastating. The aphid, because it transmits also the viruses, and this is the most dangerous thing of the aphid. And also fusarium wilt, which is present in the country, but also we have monosporescus, other pathogens that are identified also in Tunisia. But the most devastating ones are fusarium wilt, powdery mildew, and aphid. And for that reason, I focused on these three biotic stress. But also, as I said, some others pathogens exist in the countries, and our accession need to be also evaluated for these pathogens in order to have a complete information regarding the different accessions. And this will be done in collaboration with the phytopathology laboratory in our center to evaluate other pathogens in the future.
Okay, very interesting. Thanks for that one. I am just scrolling through the list of questions that we have here. You mentioned, no, maybe I take that one first, regarding melons again. The research you did perform on melons. What would you think is are the major research gaps now upcoming based on what you have done? So what are the next questions to work on based on the study you did, or the studies you did present?
In the future, what I will do, no? For the future, as I said in the concluding remarks, is we need to evaluate this accession for drought and salinity tolerance. We need to know more about these accessions and if they present any interesting other traits. For the climate change that we are facing, we have to evaluate them for drought and salinity. I think it must be done in the future, this phenotyping to abiotic stress, yes.
Right. You mentioned also, and this maybe falls together now with the last questions, the issue of plant phenotyping and the question about availability of plant phenotyping that you would need for different research activities. So from your point of view and from your practical point of view, so what do you think you would ideally need to move forward, and what do you think this is still missing, also considering what you just had in terms of drought stress tolerance investigation?
Sorry, I didn't understand very well the question.
So now you just raised the importance of plant phenotyping.
Yes.
And that's what you very well had communicated. So question being, are there any specific gaps in the availability of plant phenotyping that you would identify from your perspective? This is some technology related aspect, for example, that is missing, and where you think this would need to be available for your research?
Yeah, for the phenotyping to abiotic stress, we will need high throughput phenotyping in order to be more precise in selecting. And there is, I've see the webinars done here, and there is for the root structure and the root evolution in the soil, there is a lot of high throughput phenotyping platforms that can be used. And I think collaboration is needed here really in order to make these phenotyping to abiotic stress. I think that with the high throughput phenotyping is needed in order to make an accurate evaluation of this accession to this biotic stress, and with the platforms that exist in the different countries, this is going to help us to select probably the right accession with the drought or salinity tolerance, and this is helpful because for the abiotic stress we cannot do it by, it's done before with classical phenotyping, but with the new platforms that are available in some countries, I think it's very important to collaborate here. If someone will take me for I don't know, some collaboration here is needed in order to make such phenotyping to abiotic stress.
That's interesting you say that. I have a question here, a little bit general, but basically it asks you in what kind of collaboration you would be interested to, with other partners. So I guess this is now your chance to elaborate on that issue.
Collaboration between countries is needed also in order to make a deeper analysis and a deeper phenotyping of such phenotyping by abiotic stress.
But do you have any specific interest? Are you seeking any specific partner for collaborating further? So if you do, I guess this is now your platform to raise your interest, and we see if somebody.
In RAE, Salon, Dr. Salon has a lot of platforms and is doing architecture roots, and it's very interesting, I think. And from the webinars that I've watched, there is some interesting laboratories that we can, maybe I can do some stage training, I don't know, something like that, in order to deeper this aspect of the research. There are a lot of laboratories working on such topics, and it's very interesting to do so with the melon. I didn't find this research doing on melon crops, but another crops. But we can maybe found some interesting laboratory that can host me to do that.
Good, we've got the voice out there to that.
Yeah.
Interestingly, you just mentioned the training aspect, and sometimes we approach plant phenotyping from the technology aspect. So to what extent is this training aspect for you or for your colleagues you work an important one?
Sorry?
So is there some specific demand on training where you think this could be helpful for either for you or potentially on a broader perspective for the community?
Yeah, I think that we need all the research here because we are a lack of some facilities, and we need to make some trainings in order to make more things. For example, for the molecular markers done, it was in my laboratory in Tunisia, I was not able to do it, so I collaborate with the Spanish team in order to do the molecular markers as I did my PhD there. So I still maintain contact with my professors and with my colleagues there, and we maintain collaboration also with the Spanish team, and in all of my publications they are implicated also. So this is why, so sometimes we are a lack of some facilities or of some, to do some experiments, so we need to do some trainings and exchange between the different the European and the Tunisian country. And all the research here in Tunisia, we need that to make more deeper the analysis that we are doing. If we don't have these facilities in our country also, but if, of course, but if we have it here in Tunisia, we're gonna do it in Tunisia. But for the talking of the high throughput phenotyping, it's not well developed here. So we need to make exchange here, maybe with training. For that reason, I said collaboration is needed for the abiotic stress phenotyping using high throughput phenotyping.
Yeah, right. I'm just checking about some further questions, but it seems that your presentation and also your answers have been very specific and very helpful. the audience. Because at the moment there are no further open questions as far as I can see. So maybe to wrap it up a little bit, is there some from your side? I mean, you've got a chance now to a little bit promote maybe some of your work if you want to. So are there some publications from your side that you would advise our audience to have a deeper look?
Yes, in the bio that I've sent to Philippe, I put the most relevant publication that I had regarding Fusarium wilt, and also regarding the phenotyping evaluation using phenotyping and molecular marker approach. They are available on the website of the EPPN, I think. I have a paper that will be published soon, that I'm now making some modification, and it will be published soon. It's minor revisions. It's regarding also the Fusarium wilt resistance identified in melon accession, and the importance of the local landraces of the Tunisian germplasm in breeding program. And also I have a publication regarding the aphid resistance in the melon accession. And all of them are available on the EPPN webinar and also in my ResearchGate webpage. It's Hela Chikh-Rouhou. It's like, it is right here in my ResearchGate. Also, you will have all the information regarding all my publication in this topic.
Very good. Thanks, Hela, for that.
Thank you.
The link to the papers you just mentioned that you sent to us as organizers is already in the chat, so I can encourage all participants to have a look at these ones, as well as the ResearchGate profile that you just mentioned. I'm just going to wait for another 5 seconds and see if something comes in as a questions that we didn't miss, that we did miss so far. If not, can maybe give a very short outlook on what we're going to do next time. We're going to have obviously some more webinar series coming up, or some webinar sessions. The next one with Hamid Midipur from the Netherlands, talking on hyperslit, an ultralight hyperspectral UAV system for collecting plant spectral signature while recording sky spectra. And after Hamid, we've got Taras Pasternak from Freiburg, who will talk about plant phenotyping from whole plant to subcellular resolution. And then obviously there's some further information on our website. As Hela, you mentioned also your set of publications, which I would like to encourage our audience to have a look at for some more information. Hela, I would very much like to thank you here.
Thank you.
It has been a pleasure having you here, a pleasure talking to you. This one obviously is recorded, so on all people and the whole audience, you can have a look afterwards. And I guess, Hela, they can send you emails or contact you if there's some further questions.
Yeah, no problem. My email, you have my email, and for further questions, for further collaboration, I'm open to discussion also, proposition, all these things I will be enjoyed if I receive something.
Very good.
Also, thank you to Philippe, also for all the community, for all the audience, for watching the webinar, for the Q&A discussion. It was very exciting. Thank you very much.
You are very welcome, Hela. It was a pleasure having you and interacting with you, and I hope for the best collaborations and the best results for your future work.
Thank you, Philippe. Thank you, Sven. Thank you very much.
Okay. Thank you very much, Sven, as well. And we'll see you in about 3 weeks in our next Q&A. Thank you very much, and bye-bye.
Bye-bye. Thank you. Bye. Ciao.